Chapter 6: The Developing Brain. The first step includes formation and induction. The three layers are the ectoderm, mesoderm, and endoderm. Neural induction is when signals from mesoderm trigger some ectoderm to become nerve cells. Cell classification is an important role in this step. The fate of a cell is largely determined by its proximity to various sources of signaling molecules. For example, the signaling molecule sonic hedgehog is released from the mesoderm. Cells close to the sonic hedgehog become glial cells. Glial cells can be ependymal cells, oligodendrocyte, astrocyte, microglia, satellite cells, or Schwann cells. All of these cells help support and nourish the neurons. Cells a little farther away from the sonic hedgehog become motor neurons. Motor neurons are cells that connect the brain to the muscles. Cells even farther away become interneurons. Interneurons connect sensory neurons to motor neurons. They basically transmit signals between neurons. This picture shows brain development throughout the gestational weeks. During the third week, the human brain begins to form, and you can see the future forebrain and the future spinal cord. During the fourth week, the individual sections of the brain can be recognized. You can see the optic vessels, the forebrain, midbrain, hindbrain. During the seventh week, the eyes can also be seen, and so can the spinal cord. During 6 months, the ridges of the brain can be observed. The next step is migration. New neurons journey from the inner surface of the brain to their long-term location in the brain. The ectoderm starts to thicken, and as cells divide, a flat neural plate grows. The ridges of this neural plate fold towards each other and become the neural tube. In the picture, you can see migration in the cells in neurons in the dentate gyrus. The next step includes making connections. After neurons reach their final location, they make connections. Unlike the previous two steps, making connections is environmentally dependent. Factors such as watching a mobile spin, listening to a voice, and even proper nutrition or interacting with animals, like the baby in the picture is, help the brain to form environmental cues. Growth cones are enlargement at the tip of the axon that explores the environment. Receptors on growth cones help it to respond to environmental cues as previously mentioned. Growth cones tell the axon to go forward, change direction, or recoil. In this image, growth cones can be seen as the red color on the green axon. The last step of development is myelination and pairing back. Myelination increases speed of signal transmissions through the axon of a neuron by 100 times. Myelination causes the axons to have nodes of Ranvier. Nodes of Ranvier are spaces of axons that are not myelinated. These spaces help transmit signals faster through saltatory conduction. Saltatory conduction is responsible for rapid transmission of signals. Electrical signals skip through each of the nodes of Ranvier in order to transmit faster. Myelination occurs throughout the lifespan, except in certain neurodegenerative disorders. When we're born, we have most of our neurons. However, sometimes we have excess connections. Pairing back is important in order to create an efficient system in our nervous system. Pairing back uses the mechanism of apoptosis. Apoptosis is widely known as programmed cell death. It uses trophic factors, which are life-sustaining chemical signals, in order to detect when a neuron should die. When a neuron does not produce enough trophic factors, apoptosis begins. The nerve growth factor is an important trophic factor which is important for the survival of sensory neurons. This picture summarizes the apoptosis process. The first step is that the cell is damaged, stressed, or triggered by body signals, and apoptosis begins. In the case of development, there are axons neurons. Step 2, the cell begins to shrink and form blebs. Proteins are activated to break down the cellular components. Step 3, enzymes break down the nucleus and the cell emits signals to attract macrophages. Step 4, the cell breaks into several smaller pieces containing the small cell components and destroyed nucleus. And step 5, macrophages recognize the cell parts and remove them from the body.